Everyone, and welcome. Today we're going to continue our talk on secondary lymphoid organs. Hopefully, it'll be easier now to understand secondary lymphoid organs, having the foundation already from understanding what the follicle is and what a lymph node is. In several regards, you can think of the Peyer's patches and the spleen as modified lymph nodes. They're not, but they're all related because they are secondary lymphoid tissues. But hopefully, if you have that schema of the idea of what a cortex is and what a paracortex is and what's the B cell area, what's the T cell area, how the dendritic cells tend to be associated with T cells and the follicular dendritic cells tend to be associated with B cells, hopefully that whole schema will make these much easier to understand. Usually, the big difference between lymph nodes and the Peyer's patch and the spleen is what is the antigenic input into that structure. That's the big difference between them. So we'll harp on that, but by and large, it's pretty similar here on out. So this will be a much shorter and much easier lecture, hopefully. I also hope you're all doing well. So the Peyer's patch is very similar to a lymph node. We have about 200 of them, and they exist in the small intestine, usually in the ileum, which is the last part of the small intestine. Now recall, the small intestine's main job is nutrient absorption. But there's also some immune function here as well, and that's going to be through something called the M cell. So here we just have our columnar cells that kind of line the small intestine, right? And they just kind of form like a row, right? But like wedged between them, you're going to have something called an M cell, and the M cell is going to sit on top of a Peyer's patch, and the Peyer's patch is just a famous structure that's part of the galt. So remember how last time we spoke about secondary lymphoid tissue, there was the MALT, the mucosal-associated lymphoid tissue, and all the pretentious subcategories. Galt, gut-associated lymphoid tissue, is one of them. And so the hallmark of the galt is the Peyer's patch. So really, if you want to think about it one way, just think it's like a huge aggregate of lymphocytes. It's lymphoid tissue all under the M cell, kind of, right? This is a vast oversimplification in this drawing, but that's like a picture you can envision, right? So what happens? Let's just say that someone eats a salad or vegetables or something, hence I'm gonna use green, and then it was dirty. It had like, antigen all over it, like bacteria, and those bacteria are like shedding antigen left and right. The M cell is evolutionarily adept. That's just kind of the inherent innate system of an M cell to be able to pick up on these antigens, and it's going to package them into an endosomal vesicle, and it can transcytose them into the lymphoid tissue beneath it. So big job of the M cell, detect antigen, right? Pick it up, package it, transcytose it across itself from the lumen through itself into the lymphoid tissue beneath it, right? And what happens there? What happens to the packaged antigen in the submucosal tissue? A follicular dendritic cell will actually receive the packaged antigen from the M cell. And what happens with, I give the analogy last time of the Islamic golden ages, Bayt al-Hikmah, the great library of Baghdad and all these things. Same story here. These follicular dendritic cells that exist in the Peyer's patch get their antigen, and they're basically an antigen library, but they get their antigen via M cells, basically sending it to them. So let's just call this green star right here the follicular dendritic cell. And so if there's a follicular dendritic cell with a ton of antigen, what's going to happen? B cells are going to aggregate all around it, right? And that's how you get the basis of a follicle, light zone, and then you just have the dark zone on the outside, so darker. And that's super important. So you can appreciate like why it's important to have this. It's just basis of anatomy. Let's just say there was something picked up in your intestines, and there is antigenic stuff picked up all the time. It'll be a pain if, like, I don't know, supraclavicular lymph nodes had to like direct, their troops down towards your intestines. No, you guys want something very proximal, very regional. You could almost think of this Peyer's patch as like the small intestine's lymph nodes, kind of. They're not lymph nodes, but they kind of have that function as a big reservoir of lymphocytes. And there's going to be T cell areas associated with them as well. And so you can appreciate on the basis of there being like different areas, there's going to be like your T cell area and like the interface zone where the B cells can like do their mutual maturation with the follicular helper T cell, and the B cell can become a plasma cell. Now, on the topic of plasma cell, which is an antibody producer, it's very good to know that the T follicular helper cells and the helper T cells in this Peyer's patch uh, tend to be Th2 lineage, or the T follicular or the follicular helper T cell will have Th2 like activity. And what do we mean by that? Remember, the big function of a CD4 helper T cell is that it will produce a cytokine environment that will direct white blood cell activity, B cell activity, or B cell development for the isotype switching. Remember, we kind of made that analogy that it's kind of like whatever like the T cell makes the broth, and the broth is the cytokines, and then the it's like hot pot, right? The B cells like the meat, and then whatever broth you cook the meat in, that's the flavor it will get, and that's like the isotype switching it will take. So if you have these Th2, and remember, we also categorized them from the earlier lecture on like what type of cytokines each subset of helper T cell produces. Th2, remember, are the IL-4 and 5 and 13 producers. And remember, IL-4 is very good for IgE production. So a B cell who wants to go isotype switching, who's smothered in IL-4, will become an IgE producer. If it's smothered in IL-5, it will become an IgA producer. And then IL-13 is just like a pro-mucosal agent. It doesn't really do too much with the isotype switching. It just basically makes mucosa more active. And that's important, because if you have a lot of mucus getting cranked out in your GI tract, it's going to be filled with mucins, and the mucins are these big carbon structures that are really good at capturing viruses and bacteria, right? And so remember, function of IgE has a big role in like intestinal infections and parasites, and then IgA is like your hallmark mucosal antibody. It tends to be a dimer. It does a really good job. Tends to have like very good affinity and everything. So it's very good to have IgE and IgA in abundance in your alimentary canal. Unfortunately, the lymph nodes, like the lymphoid tissue there, knows that, so that's why it's gonna emphasize that pattern of growth for the T cells down there, to produce B cells that can produce that kind of antibody. It's all full circle, right? All full circle. So we're clear here, and then we'll move on to a picture. Yeah, so this is what it looks like. So you have your intestinal cells, right? And then right below them, like submucosal type area, you just have this big aggregate of lymphoid tissue. That's going to be a Peyer's patch. The spleen is a beautiful organ, and the next organ we're going to talk about. It is kind of like our body's blood filter. The spleen is very remarkable. It exists in your left upper quadrant of your abdomen. It has direct contact with your diaphragm, and that means if you irritate your spleen via the phrenic nerve, which innervates the diaphragm, you can actually get a referred pain pattern to your left supraclavicular area, which is basically just your left anterior shoulder. So you irritate your spleen, you get a referred pain pattern via the phrenic nerve to your left shoulder. It's called Kearsign. It's I guess one of those clinical things. But the spleen, early on in development, is also hematopoietic, so it actually produces blood cells like our bone marrow does. In an adult, though, you really don't want a hematopoietic spleen. That usually might mean you might have something funky going on in your bone marrow, and that's not a good thing, right? But aside from these things, in the normal adult human, the spleen is basically a very vascular organ that acts as a filter and as a secondary lymphoid tissue. So there's going to be some antibody production there as well. So what sets it apart from a lymph node? Spleen, by the way, is about 6 centimeters, about the size of your fist. Why isn't it just considered like a giant lymph node? Well, remember, it's not a lymph node. It's different. It has other things too. Your lymph nodes don't produce red blood cells. They never have. But the spleen is really the way you want to think about it, right? It's going to have like a very specific subset of macrophages who basically act as filters. Like they basically like scanning the blood, and they eat problematic things in there. But also you want to think about it as a place where there is something called marginal zone B cell, and this is a specific subtype of B cell that has like a very niche function because it doesn't need co-stimulatory signal to become an antibody producer. And so we'll talk about that in a minute. But just appreciate what the spleen can do. There's also something called the periarteriolar lymphoid sheath called the PALS. This is going to be like your T cell storage of your or T cell area of your spleen. So let's I kind of said the same thing here. Let's draw out the general schema of the spleen. Okay. So the way you want to think about it, right? You're gonna have your splenic. Well, first, anatomy. Off your celiac trunk, which comes off your aorta, you're gonna get the splenic artery. Splenic artery is going to really give out like these cords, and these cords are kind of percolate through the spleen tissue, and they'll end up in these sinuses. And the sinuses will basically go out to this splenic vein, and then from the splenic vein, the blood will get dumped into the portal circulation. And since we've come full circle, so off splenic artery, we're gonna start. So we have the arteries, and then off those you get like these little arterioles. Okay, so these arterioles, they're gonna be the basis of basically attracting antigen, or how antigen gets in. So a big distinguishing point between a lymph node and the spleen is that the spleen doesn't have lymphatic afferent vessels. That's a critical point. Antigen only gets to the spleen via the blood. There is no input lymphatic vessels into the spleen, right? As opposed to the lymph node has both. But spleen only has blood supply, right? So we can call this whole area the white pulp. So we'll call that white pulp. And then you're going to have something called the marginal zone. We can call this MZ. And then you're going to have something called the red pulp. And we'll call this. From marginal zone, from the marginal zone, you're actually going to have a continuation of the arterial flow. And then once you get to the red pulp, you're going to have the formation of like these sinuses. And so I'm going to do that one purple, because the sinus, it's just basically going to be like a connection of venous and arterial blood. And so we have to establish that the arterial flow is going that way. And I don't know if you're how familiar you are with grasshopper anatomy, but this kind of reminds me of that. Grasshoppers have something called an open circulation. Their blood and their lymphoid tissue is actually combined. It's called hemolymph. And the way it works in a grasshopper is that basically the heart pumps this hemolymph into like the cavity, like the body cavity of the grasshopper, and then blood just basically sloshing up across like the internal structures, and that's how it gets its blood supply, and then that's basically reabsorbed and then kind of sent back to the heart. It's opposed to like a closed circulation that like humans have, that has like a formal capillary formation and things of this nature. Nah, grasshoppers is just like a big empty vessel, and then hemolymph is sloshing in, and it's like the contractility movements of the grasshopper that kind of can help usher hemolymph back to the heart. But I don't know, the sinus of the spleen kind of reminds me of that a bit. So what's significant here? If we can take this area, and so that's basically how the blood supply happens. So if you're going from arterial to venous, and we have something happening in the white pulp with regards to like the formation of these follicles, let's zoom in. What does the follicle entail for this? So this is like the main branches of splenic artery. So then they form like these little arterioles, right? So surrounding the arteriole is actually going to be the first thing we spoke about, and that's going to be the periarteriolar lymphoid sheath. So this is the PALS. And so this is going to be your ugly A. Wow, look at that handwriting. So the PALS, this is going to be your T cell area, right? And again, I don't know if I mentioned this mnemonic already. It's nice that paracortex is T cell area and PALS is T cell area, and both PALS and paracortex start with PA. That was nice, right? And then if there's a T cell area, I hope you can appreciate that this is going to be the B cell area, right? And so that means here we have a B cell area, T cell area interface, right? So this is a follicle will basically form here, and then there's a T cell area, and when the B cells find their antigen, and they're going to do their other somatic hypermutation and clonal selection, all these things, they have an interface with T cells to work for, right? And then you have like macrophages and dendritic cells interspersed who will actually be helping with the antigen collection and sampling. So this is the basic schema of what's happening in this. This is just like one unit, if you will, but this is happening in the spleen. And so now you have to like beg the question: what's happening in the marginal zone? So the marginal zone is actually going to have some macrophages who are like literally perched up along the vessels, looking for something dangerous to clear it. That's innate immunity. And then you're gonna have these little pockets of things, and I don't know what color I'll do them in. I'll do pink. They're gonna be called marginal zone B cells. And so marginal zone B cells is what we're gonna talk about next. So marginal zone B cells are B cells that don't need T cell help to become antibody producers, kind of like a plasma cell function. Why do they exist? Well, it's because of the nature of bacteria and the nature of our immune system. We're going very to the nature, okay? Remember, this whole adaptive immune system story is dealing with protein antigens. Bacteria, unfortunately, are very good at having non-protein antigens. They have polysaccharide antigen, and polysaccharide is a sugar, right? So sugar antigen is a thing. So all these mechanisms in place that we've been talking about that are adept to dealing with a protein antigen are rendered near useless if confronted with a sugar antigen. And unfortunately, that's like what bacteria love to do. They love to have sugar antigens. So what's the workaround? Resign ourselves to the bleakness of this reality and we all die? No, humans would have gone extinct a long time ago. We were created with the marginal zone B cell, and it is a lovely thing. It doesn't need a co-stimulatory signal with the helper T cell in order to activate and become an antibody producer. Rather, just through this marginal zone B cell having a toll-like receptor, much like a lot of your innate immune system cells, like the dendritic cell and the macrophage, through a toll-like receptor and abundant cross-linking of the receptors it does have, it can activate on its own. This is a very unique phenomenon. And they will form generally IgM, but they can also form IgG and IgA. But it's good because IgM and IgG are very good opsonins. These are generally the two considered. These two antibody classes are considered to be like the opsonization class antibodies in this context. And also they can both serve as a tethering point for a complement system, right? From way back when. So marginal zone B cell, I want you to think of T cell independent activation, and the utility is it can work against polysaccharide antigen, right? So this is very important. So why do we run into polysaccharide antigen? That's because many bacteria have something called a capsule. Capsule is going to it's basically a complex of water and sugar, and it basically slime coats almost the outer cell wall of the bacteria. It's a virulence factor. That means it can help evading with phagocytosis and basically make that bacteria more spreadable, which is scary. And it's very similar to something called the glycocalyx, which is even similar in structure. But the thing about the glycocalyx, that's mainly for adhesion. and it's not really so much a virulence factor. As opposed to the capsule, it can do adhesion, but also phagocyte evasion. The way I like to think about it is like it's like covered in soap, and then the macrophage is a hand that goes to grab it, and then it can basically slip out of the macrophage's hand because it's like covered in soap. That's the way I kind of envision that. The big way that the body clears encapsulated organisms is through an IgM response or IgG2, which is a subclass of IgG. That's a big way it's cleared. So you can appreciate that if the part of the body adept to dealing with polysaccharide antigen is impaired, you have an increased risk of infection with bacteria that have polysaccharide capsules, right? And this is seen in asplenia or functional asplenia, because spleen not working is going to predispose to this, right? And so usually you can see this in like some cases like sickle cell disease. If it progresses far enough, you can actually get a functional asplenia. It's quite common. So in that case, vaccination against encapsulated organisms becomes super important. There is a unfortunate correlation between strep pneumo, Streptococcus pneumoniae, caused sepsis, and asplenic individuals with sickle cell. Strep pneumo is a very prevalent encapsulated gram-positive organism. But yeah, that's just the thing to get straight. No splenic macrophages to filter the blood, and no marginal zone B cells means decreased immunity in the blood. I mentioned last time that after I finished the immunology series, I wanted to get into like some microbiology and specialized immunotopics. This is just like what's to come, I guess, but this is just like a dirty list of encapsulated organisms. And the only three I'll really talk about right now are Neisseria meningitidis, which is your it's a gram-negative, very infamous, causing meningitis. And you have Haemophilus influenza type B. This is a gram-negative coccobacillus. It is there's a very known vaccine against it, and then strep pneumoniae, which we spoke about earlier. These three are all encapsulated, and they all cause meningitis. These aren't the only three that cause meningitis. You have others, but these three are like three sisters. I tend to call them. They're very famous. They're very famous causes of meningitis, and they all have capsules. So you can actually imagine anyone with this sort of asplenia has risk for these three organisms causing them a problem, because they're encapsulated. There's another association like salmonella and sickle cell individual patients. That's also a well-studied one, and you can appreciate it's just not a rote fact to memorize, but if you could kind of appreciate, oh, salmonella is also encapsulated, and then sickle cell might have a spleen problem, you can kind of make that correlation. Yeah. And like I said before, the spleen, the capsule is usually just polysaccharide and water. But there is one exception. It has nothing to do with today's talk, but it's anthrax, Bacillus anthracis. That actually has a capsule made of D-glutamate. So it's a little bit different in structure than another capsule. It has nothing to do with today, but that's just like a commonly tested thing. Like the clinical vignette will describe anthrax, and it's like, what did this? And it'll just give like characteristics of bacteria that are kind of vague. But if you can just recognize the characteristic as, D-glutamate, you can usually get the question right. That's kind of like the schema of studying I won't get into it too much right now, but the schema of studying microbiology is just how you can call one thing by many different names. So very seldom will the question just straight out ask you what's the bacteria. No, it'll describe the bacteria, and it'll ask you to give one aspect of it. So it's kind of like web learning, that's just something else. But yeah, making that bacterial capsule infection or encapsulated bacteria infection risk with asplenia is very important to be able to make. But hopefully by understanding the immunology with regards to splenic macrophages and marginal zone B cells, you can appreciate why that association exists. That's the whole point of this section of the video. So as we wrap up lymphocyte trafficking, naive T cells, before they are like ripe to enter the rest of the body, their surface markers kind of confine them only to the lymphoid tissue. That makes sense because you don't want a useless T cell out in the circulation. It won't be able to do much. You kind of want it to make sure that it gets its dendritic cell type help and it can become an effector, CD8, and all these things. And same story with the helper T cells as well. But mind you, follicular helper T cells, they're a specific subclass of CD4. Those tend to stay in the lymph node or the lymphoid tissue as opposed to go out in the circulation. But I guess no, usually. Lymphocyte trafficking is a phenomenon in which if a T cell or a B cell, for that matter, isn't adept at its job yet, it won't be sent out to the site of infection unless it's ready. And again, some cells, like follicular helper T cell or some plasma cells, don't really leave their initial area because they can just kind of do their job where they are. Like, for example, some plasma cells will just hang out in the lymph node and they just crank out. They'll just crank out antibodies from there. Others will go to the tissue. But it's because remember, it's not the plasma itself doesn't kill. It's the antibodies that it produces that will help other things kill that thing, right? Because antibody can, if it's IgM and G, it can fix a complement, or it can act as an opsonine, or it can neutralize a virus. Things of this nature, right? Things of this nature. So I hope this was able to help you kind of conceptualize secondary lymphoid tissue. That's pretty much the big ones to know. And again, with regards to the MALT, I really just spoke about BALT, and because of the Peyer's patch, which is a very well-studied example, and then spleen, and then your lymph nodes, right? So these are my sources. I'll see you guys in the next one. Take care.